Hopefully everybody can hear us online now. It's my huge honour and pleasure to introduce Professor Bob Turner, who's a neuroscientist, physicist, social anthropologist in his career. I hasten to say the word, one of the grandfathers of MRI, maybe one of the fathers of MRI, who's made a huge contribution over many years. Actively shielded gradients, diffusion imaging work with Danila Bahan in NIH, bold contrast there. Some of the first activation studies with the late Jack Vellevo. So Bob is really impeccable MRI credentials. We're very honoured indeed. I mean, fortunately, he lives in Cambridge, but still maintains his academic interests at Kubrick in Cardiff and the Sir Peter Mansfield unit in Nottingham. So, Bob, please tell us about the Myelin Bootstrap and new applications of MRI.
I have to start my talk by doing up my shoe. I also need some way of moving these slides along. So
Hopefully, it should just be
maybe the down arrow. No, it might be the. That's it.
That's the one there.
Going.
Right.
Well, I'd like to start by thanking Freddie and Martin for inviting me to give a talk here. It's not often I get invited to give a talk at Addenbrooke's, even though it's just up the road. Though I have interacted quite a bit with the neuroscientists who work here, and other people who use the scanning facilities. But what I want to do this evening, I should say, is to talk about something that has become increasingly fascinating for me over the last few years, which relates very much to some of the motivations which I had back in the 1990s for going to very high field MRI, which I did in those days with Roger Ordage in London. But things really have moved on, and you'll find out how as I give the talk, which I'm going to go through rather quickly because I have rather a lot of slides, but I think you'll find them interesting. So, first of all, I just want to point out that MRI is not just a diagnostic modality. It's wrong, terrible to me to say this to radiologists, but actually with the discovery science methodology as well, we actually can discover something about our brains, for instance, using MRI, which nobody knew before. And as partly because of the push of that hope of discovery science, qualitative imaging, which had been the kind of the hallmark of 1980s radiology with MRI, has evolved into quantitative imaging, first of all in the quantifying the block parameters, but then later Dennis Levihan and I got water diffusion mapping clinically feasible using echoplanar imaging and cerebral blood burn volume, and then most latterly myelin. In my talk, I want to emphasize the importance of neuroanatomy, in particular the bands of Bioget. How many people in this room know the term bands of Bioget? Okay. The most important feature of the human.
I knew a radiologist.
Just to point out that there are about 360 cortical areas, 180 in each hemisphere, and more than 400 named deep brain nuclei. And the question is, what do they each do? I think it's the question that radiologists should probably be interested in as well. So just a blast from the past. This is what we published in 1990. The first decent artifact-free images at 1.5 Tesla with differential diffusion weighting from the top left and bottom right, using a home-built head gradient coil, which I designed and had built at the time. So what can in vivo MRI tell us about neuroanatomy? On the right of this slide here, we have a section of cadaver brain stained for myelin. On the left, we have a map of T1. It's unavoidable that we can interpret T1 as a map of myelin. And I'll say much more about this later on. But I want to mention a few more topics that I'm going to bring in. One topic is brain maturation. Another topic is excitatory and inhibitory microcircuits and the normal neuronal oscillation spectrum in the brain. I'm going to mention pyramidal cells and basket cells. I'm going to now mention, this is the magic word that I mentioned to Martin, parvalbumin. How many people in this room know about parvalbumin? All right. And keep this question in your heads. Are the bands of Piaget actually the major inhibitory axonal pathways? Now I'm going to talk about MRI and myelin quantification, and myelin sensitive MRI as a specific biomarker for mental illness. So we know that experience shapes the brain, and these are three studies which were fairly important in this development. The first one was done with electrode recording in the cat brain, where they found that if you lesion the hand area in the primary sensory cortex, it resulted in the reorganization of the secondary hand area in S1, S2. So the brain reorganizes itself in response to lesions. And then there was a paper by Avikani et al. This is in the lab of Leslie Angerleider, the first longitudinal fMRI study, which I happen to be the MRI physicist involved in. And this showed that if you practice a particular finger tapping task, you actually increase the area of primary motor cortex which supports that skill. And then there was a work done by Bragansky, this was in London, 2004, which is the first longitudinal structural MRI study showing that the grey matter density in V5, the visual motor area, the visual motion area, the grey matter density changes after 3 months of juggling practice. So all of these things are telling us that the brain is highly plastic in normal subjects. Just going to mention now this technique for mapping T1 quantitatively, which we published in 2021. This is with a group of Sue Francis in Nottingham, which uses a two-dimensional multi-shot, so single slice, multi-shot inversion recovery EPI sequence, which gives very high resolution, very high signal to noise per unit time, and very precise T1, values of T1. And it's basically more efficient than the MP2 range sequence which people use for mapping T1 now. It's still not been implemented properly on a Siemens system. This was implemented on a Philips 7 Tesla system. But it has inherently better resolution than MP2 range. It has a tighter point spread function. So it's a sequence which I'm quite excited about. A highly efficient sequence. But with this sequence, you can pick out layer structure in the cortex in these 0.5 millimeter isotropic scans, which didn't take very long to collect. But what do we get when we have myelin? Well, the question is, does T1 really give us myelin? And this is now a study which was done by my student Carsten Stuber and me and collaborators in my lab in Leipzig, which demonstrated that iron and myelin were the two main contributors to all brain MRI. So we performed ex vivo MRI scanning of cadaver brain, and then we sliced it, and then we gave adjacent slices various histological stains. One slice out of each bunch was analyzed using a proton beam accelerator, so-called Lixion. But what we see in this slide is a stain for myelin-based protein. This is primary visual cortex. And then the iron map corresponding to it, and then the pearl stain for iron. The iron map comes from the proton beam accelerator. I'll mention how that works. So you fire protons at the tissue, and then you get characteristic X-rays coming off. You get a spectrum of X-rays, and the peaks in this spectrum quantitatively reveal what elements are actually present in the sample. This paper has now been cited hundreds of times because it was the first of its kind. So the iron beam runs through the sample line by line. Now, it so happens that iron is vitally important for the formation of myelin. It is a cofactor. Oligodendrocytes, which are the cells which form myelin, are the predominant iron-containing cells in the brain. Not counting red blood cells. The iron is found in the pericardium carrion of oligodendrocytes, and it's required for the onset of myelination. It's mostly bound to ferritin and mineralized iron oxide. Iron and myelin go hand in hand. So what Carsten Stuber was able to do was make quantitative myelin maps, because he was able to measure the phosphorus, P, and sulfur. And there's a standard quantity of phosphorus and sulfur in myelin, in the lipids that form myelin. And so by comparing the levels of phosphorus and sulfur, he was able to actually make maps of the myelin. And when he did this, he was able to compare the value of 1 over T1, that's R1, with the iron and myelin in this very decent sized sample of brain tissue. And he found that there's a beautiful linear relationship between R1 and the concentration of myelin. So that's a good way of getting at myelin, measuring T1. So what is myelin? It's the most common substance in brain tissue. It's what wraps axons. The components were first identified by Ludwig Tüdicke in 1884. He worked in London in his garden shed, boiling up cow brains, distilling them. And it's quite striking that about 27% of the myelin lipid is actually cholesterol. Now, you might be surprised to know how much cholesterol we actually need. It stiffens the membrane. And then there's all these others. He gave the name to several of these components. He invented the names like sphingosine and so on. These are the membrane proteins. So moving on now, about 150 years, to work done by Douglas Fields at NIH. He was able to show that myelination is largely driven not by time or age, but by activity. So we myelinate our axons by using them. there's a bootstrap mechanism, which has been well described by David Myelin and now others, in which electrical stimulation of axons causes release of glutamate from vesicles activating the NMDA receptor and the metabotropic glutamate receptor on oligodendrocyte cell processes. That triggers formation of the axoglial signaling complex. We can read that out. But the activated FYN phosphorylates the heterogeneous ribonuclear protein A2, resulting in local myelin basic protein translation from mRNA in oligodendrocyte cell processes. New membrane is formed and wraps axons. So we have a mechanism, a pathway, why myelin is bootstrapped in the brain. For those of you who know or are interested in MR spectroscopy, it turns out that one of the big reasons we have all this N-acetyl-aspartate, which is a big peak in the proton spectrum of brain, is because it's actually used in promotion of myelin synthesis and energy production in oligodendrocytes. It has a vital role in the formation of myelin. So this is a neonate cadaver human brain from the work of Paul Plexi, who is the father of the study of myelin in human brain. And you can see just how little myelin there is in our brains when we're born. It's only basically in the corticospinal tract and in the optic tract. And in the cerebellum, obviously, you see quite a lot. But elsewhere, basically no myelin. Now, it's been shown that the axonal fascicles, the fiber bundles in white matter myelinate sequentially. And this is work of Brody and Kinney in neonatal and infant myelination with the cadaver brain studies. It's also been shown that T1 varies between white matter fascicles quite recently, while it remains relatively uniform within each fascicle. So each fascicle has a distinguishable longitudinal time course over the lifespan. Starts before birth, and these are in Nature Communication 2022. These are our maps of myelin in newborn, 3 months, 6 months, in different fascicles. They use the diffusion-weighted tractography, obviously, to calculate the fascicles, and they look at the R1, the relaxation rate in newborns, and each of these points along here. Is there an arrow that I can use somehow? Yeah, there is. Each of these points represents a different fascicle, like the optic tract. And the CS is the cerebrospinal tract. And you can then look at the rate at which they myelinate, and they all myelinate at different rates. The grey matter also myelinates sequentially, and it must be related to behavioral acquisitions and development of cognitive abilities. So if we're looking at the myelin, we're looking at a map of what's working, what's effective. And so that's the work of Levenberg. So where does the myelin go? Well, it goes into these, oops, get that back. This is a myelin stain section from my lab in Leipzig. And you can see nicely that obviously it's in the white matter. It is also very typically in 2 layers within the grey matter. These are called the bands of biochain. And also there is a layer of myelinated axons at the surface of the brain in what's called the molecular layer, or layer 1 in the cytoarchitectural terminology. So what's it doing? Why is it there? Why does it come in layers? And more particularly, why do those layers vary from one cortical area to the other? Elliot Smith, who was a neurosurgeon at the beginning of last century, in Cairo actually, studied the myeloarchitecture of cadaver brains that he found very easy to get hold of in Cairo. And he said there is a very widespread belief that the characters of one area merge gradually and imperceptibly into those of neighboring areas, but this is entirely mistaken. The changes in structure occur with the utmost abruptness, so that it is possible to determine with absolute precision the exact boundaries of each area. If the reader needs convincing the accuracy of the statement, let him cut a fresh brain at right angle to the calcarine, intraparietal, any part, central, inferior, frontal, or parallel sulci, and he will find all the confirmation necessary. And it is striking when you start looking at stained sections of brains just how clear cut these boundaries actually are. This is a high resolution so this intersection of human cortex showing these radial fibers coming out from the white matter and the horizontal fibers, which don't always obviously lie in the plane of the section. In fact, generally speaking, they don't. So you can see little bits of them, but you can see how they are more or less at right angles to the radial fibers. So there's parallel universe in looking at brain anatomy besides the calculation of the cortex due to Brodmann, which is the one that most people use using the cells, the neuron stain. We have the myeloarchitectural map. This is one by Elliot Smith in 1907, which match rather well with each other, actually. The great thing about the myelin maps is that we can see them with MRI, because MRI, as I was saying, is the main source of contrast in the brain. And we were doing this in Leipzig. We were mapping the T1 at 3 separate equivalent contours within the thickness of the cortex. And so at different depths in the cortex, we get different maps of the myelin, which again match up with the work using a microscope of Elliot Smith. So you'll see the primary motor cortex very heavily myelinated and the primary visual cortex very heavily myelinated. So that's we used 0.5 millimeter maps of T1 to do that, and using Miriam Baynett's beautiful software, we were able to construct 21 laminae between the white matter surface and the field surface. And David Van Esten and Matt Glasser got onto this idea very quickly. They learned about it at a human brain mapping meeting in Barcelona, where I was presenting my posters on this topic. And they got onto this very quickly, and by 2016, they made this beautiful map of 180 brain areas, which is similar to the myeloarchitectural map of 1919, which I showed on the previous. No, I didn't show that, but it is similar. The comparison between the grand myeloarchitectural map of Oscar and Cecilia Bach, published in 1919, and the Glasser and Van Esten map of 2016 was made just in a paper just published. This is the final and last paper of Rudolf Neuenhuis, the great Dutch neuroanatomist who just died a couple of months ago. This was his last paper. I had a great honor and privilege of working with him for several years, based in his home near Amsterdam. Anyway, he and Matt Glasser got together and actually made a comparison of their respective maps of the brain. This map was based on the work of the Oscar and Cecile Bach over 50 years in Germany. And of course, this is the map that was made using functional connectivity and a variable which has been called T1 over T2, which it isn't, but that's its name. Just wanted to say about these myelinated fibers and talk about the tangential connection. Myelinated axons within the cortex are radial or tangential, and there's three classical types of tangential fibers. There's the U fibers, which are just inside the white matter, and they're coherent in direction. Then there's the bands of Baillet, which I just mentioned, which characterize cortical areas. They're incoherent in direction, and they terminate at cortical area boundaries, very interestingly. And then there's a layer in the surface, which is sometimes known as the excellent stripe, and that happens to be coherent in direction as well. And we showed that in cadaver brain sample like Christoph Leutz and my PhD student. But this is the crux of it, that work by Micheva published in 2016 in I think mouse brain. She showed that the intracortical tangential myelinated fibers are mostly inhibitory, and they arise from basket cells. They're GABAergic. We don't know about the other tangential connections, whether they're inhibitory or excitatory, but having a handle on the inhibitory microcircuitry of the cortex gives us a huge step in the direction of actually understanding what the heck it does. So, what does myelin do? There's the following 5 properties. It accelerates the propagation of action potential by a factor of up to 100, decreases the energy cost of transmission by an order of magnitude, it makes axons stronger, it reduces crosstalk, it reduces synaptic plasticity by inhibiting neurite growth. So it casts areas of brain cortex in stone. Myelination of unmyelinated axons can be promoted by electrical activity, as I was saying. Axonal myelination is the final stage of neuronal maturation. Myelinated axons are privileged pathways for action potential. So can reverse engineering analysis of myelin architecture help to explain the structure function relation of cortical areas? This is a $1,000 question. In my recent study of the literature, I'm happy to propose further functions of the transverse cortical myelination, the horizontal or tangential myelination. I claim that it prevents seizures. It provides area-specific collective suppression of cortical activity, such as fast spike of V1 cortex via the stream of Gennari, to avoid visual motion blur. It sculpts population receptive fields, such as center surround inhibition. And it generates identifiable temporal patterns of spiking activity and hence frequency spectra, which we can measure with EEG. So it's kind of important if those are, or if they turn out to be the case. Myelination, as I say, is driven by a neuron's experience. It's quite uniform within a cortical area. Does a shared pattern of myelination imply shared experience? If so, we can ask whether each anatomically definable cortical area has a specific set of computational competence. Are these stages in motor and cognitive development reflected in episodes of more rapid myelination of specific cortical areas? As the axons myelinate and neurons come on stream and become dramatically more effective participants in brain-wide neural networks. So here's another slide of statement. Human brain axons are progressively myelinated, driven in a bootstrapping process. Distinguishable cortical areas have different patterns of myelination. They define the most important microcircuits in each area. The horizontal myelinated fibers usually arise from fast-firing parvalbumin inhibitory neurons, basket cells. I'll say much more about that later, and carry GABAergic inhibitory signals. Brain MRI contrast largely depends on the myelin content in each voxel. Thus, in vivo, high field MR brain images with spatial resolution sufficient to distinguish and quantify myeloid architecture should provide uniquely valuable insights into human brain development and function, coupled with high-resolution MR tractography and line-dependent MRI. General population-wide trends in myelogenesis during development and individual differences should become key explanatory observations for cognitive psychology and the basis of empirical biomarkers in psychiatric disorders. There's a flash image taken by Robin Heidemann, who's now the boss of vacuum MRI at Siemens. It's 360 micron isotropic resolution. It was taken in 2009. It's a long time ago. So now I'm going to talk about parvalbumin. Many neurons have myeloid axons. There are two main types of neurons: pyramidal cells, which are excitatory, and basket cells, which are inhibitory. Pattern of myelination defines each cortical area. Pyramidal cells mostly have radial axons, and basket cells have mostly tangential axons. Myelin is the main source of MRI contrast. Driven by experience, basket cells contain parvalbumin and GABA. They sharpen parvalbumin stained pathological sections. And there we are. Here we have the sections, basically the same section, adjacent sections in the cadaver human brain. That's the, oops. That's the myelin stain on the left. You can see the sphere of Gennari here, defining the primary visual cortex. And here's the parvalbumin stain. I'll say more about that in a bit. First of all, I need to tell you about what is parvalbumin. It's a calcium-binding protein. The parvalbumin positive neurons are the largest class of GABAergic inhibitory neurons in the central nervous system. It's really, really important. They're fast spiking. They use a lot of energy. They provide feed-forward and feedback synaptic inhibition onto a diverse set of cell types. They basically control the activity of the pyramidal cells. They're crucial for regulating sensory plasticity. And myelination is required for the establishment and maintenance of powerful fast spiking interneuron-mediated feed-forward inhibition of cortical circuits. And most parvalbumin neurons are basket cells or chandelier cells, but it's basket cells that do the heavy lifting for intracortical inhibition. So these are facts that one should understand if one understands the cortex at all. These are important facts. Parvalbumin and cognition. In EEG recordings, frequencies above 40 Hz correlate with cognitive activity. Schizophrenia diagnosis correlates with the depletion of parvalbumin interneurons and a reduction in gamma band activity. Spatially localized abnormalities in cortical myelination have been noted with MRI in subjects with bipolar disorder, autism, and schizophrenia. But these images did not have enough resolution to distinguish between radial and horizontal myelination. The question is whether 7 Tesla MRI can identify these disorders in vivo. I'm now going to show a few slides sort of underlining what I've just been saying. This is work by Lardam et al., published 2009. If you drive fast spiking cells, you can get gamma rhythm and can control sensory responses. This is in mouse barrel cortex in vivo. The experimenters targeted optic genetic manipulation selectively to fast spiking interneurons, that's the parvalbumin interneurons, at varying frequency. They found that light-driven activation of these neurons selectively amplifies gamma oscillations. In contrast, pyramidal cell neuron activation amplifies only lower frequency oscillations. So these data directly support the fast spiking gamma hypothesis. The first causal evidence of distinct network activity states can be induced in vivo by cell type specific activation, in this case, parvalbumin cells. Here's a paper on parvalbumin interneuron deficits in schizophrenia. And this is by Oscar Moran, 2024. Parvalbumin interneurons are critically involved in generating and maintaining cortical rhythms in the gamma frequency, which are essential for complex cognitive functions. Deficits in parvalbumin interneurons have been frequently reported in post-mortem studies of schizophrenia patients, and alteration of gamma oscillation is a common electrophysiological feature of the disease. So the question is, can we infer dominant cortical microcircuit by cortical area from myeloarchitecture and new fibers? Could the horizontal myelinated connection be crucial in defining population receptor fields? Are the bands of Bayer the axons of basket cells? If so, have we been staring at the missing components of cortical circuits for more than a century without recognizing them? So this is another paper by Stedehauer et al. Lab in Rotterdam, I believe. So this is again, they're looking at what happens to these parvalbumin cells when you drive them. In this case, he uses chemogenetic stimulation in the medial prefrontal cortex in, I think it was rat brain. It significantly enhanced axonal myelination. You can actually see it happening in vivo. And he shows pictures of what happens, how much more myelin, which is in green, you get on these basket cell axons when you stimulate. Myelination synchronizes cortical oscillations by consolidating parvalbumin-mediated phasic inhibition. So what they did here was they had a mouse, they removed the myelin by using cupresone. If you feed cupresone to any animal, it destroys the cortical myelin. However, it preserves the parvalbumin-positive interneurons, reducing their excitability. But if you lose that myelin, you get interictal spikes and behavioral state-dependent amplification of theta rhythms. And it abolishes gamma oscillation. So the cellular microarchitecture of myelination of the axon of the parvalbumin basket cell is shown to be required in quiet wakefulness for generating stimulus-induced past gamma frequencies and limiting the power of slow cortical oscillations and interictal spike. It really, really is important. Now, it turns out that quite recently, in 2022, Annika Alkemade, who I've collaborated with in Amsterdam over the years, and her group have produced this wonderful 3D map of microscopic architecture and MRI of the human brain. It's a concordance map where they used many different stains, all properly co-registered and co-registered to the MRI scans. This is a wonderful set of images of the brain to plow us through and try to understand. I particularly got interested in the cortical myelin and parvalbumin maps. So I've been preparing the histological sections stained for each of these. That's a sort of generalized picture. You can see on here that they've stained for calbindin, calretinin, parvalbumin. There's a silver stain, that's for myelin, a nickel stain for cell bodies. And they've been able to line these all up. Now, what I found was that the parvalbumin stain is fairly uniform across cortical areas. It doesn't vary that much between different cortical areas, but the myelin stain is less uniform. There tends to be one very broad diffuse band of parvalbumin, but the myelin can lie in quite sharp layers. If you blow up these sections, you can actually see these parvalbumin neurons. There are a few microns across, and you can see some of their axons in the parvalbumin stain. They're rather hard to see the axons, but you can see what must be the same axons in the myelin stain. These are going from left to right, top left to bottom right. Those are the radial axons. And at right angles to those, you can see some of the tangential axons in the sinus lesion. These sections are 200 microns thick, which is why they look kind of blurry, because you can only get see what's happening at a particular focal depth. Everything else is blurred out. But it's very clear from these maps that you can see, for instance, primary auditory cortex is heavily myelinated. That's A1 there. You can see that there's a greater density of parvalbumin in exactly the same place. The primary visual cortex has the stria genari, which you can see here. And in the parvalbumin stain, it's also a higher density of parvalbumin. When you blow that up, you can see it has even an extra layer of parvalbumin cells, which corresponds very nicely to the stria genari in the myelin stain. So you see two layers of parvalbumin cells in the visual cortex. This is now a human brain. And these pictures give us more questions than we can actually address yet. How should we measure myelin in vivo with my MRI? Well, I'm arguing that one should not use the ratio T1 weighted and T2 weighted. By the way, people who think that T2 weighted brain images are actually T2 weighted need to think again. They're actually T1 weighted and magnetization transfer weighted, but they're not T2 weighted. If you make maps of quantitative T2 in the brain, there's almost no contrast between grey and white matter. On average, they have almost exactly the same values of T2. So calling those maps of those images of the brain T2 weighted is actually factually incorrect. But it is traditional. And people use this ratio of T1 weighted and T2 weighted, which are usually turbospin echo images, as a kind of arbitrary index of myelination, but they're not really quantitative. They're not a physical quantity. But T1 is. T1 and MT have been shown to be more reproducible. So that's that section. Now I'm going to talk about what are the uses, new uses of MRI, which I mentioned in the title. This is a list of the global burden of disease, and what pops out is neuropsychiatric conditions, which is about 20% of, accounts for about 20% of disability adjusted life years. That's huge. But when we look at how much money is spent proportionately on these different conditions, the burden of neuropsychiatric disorders and percentage of all disorders is, say, around 13%. But the budget as a percentage of total government health spending is about 2%. So it's a very poor relation of physical health. And there's a reason for that. It's because we don't know how to define it properly, and we don't know how to cure it. Define categories of mental disorders are ontologically problematic on three levels. They're abstract entities, mental disorders. They're not clearly natural processes independent of the imposition of values or human interpretation. It's not clear whether mental disorders exist in the world apart from the individual persons who experience them and thus instantiate. Which is why the NIMH in 2013 decided to rule out the use of DSM categories of mental illness and instead put in a requirement for stating this research domain criteria, so that they're talking about symptoms rather than categories like schizophrenia and so on. There are serious problems with current drug treatments, everybody knows. Antidepressants often have the effect of suicide, far too often. For several disorders, cognitive behavior therapy works better. So what goes on there? I don't know. Any human skill that takes more than a day to learn creates a measurable trace in fresh myelin. Can we find biomarkers for mental disorder? Can we find them in the brain? That's a challenge. Psychiatric disorders are usually disorders of thinking. Symptoms develop slowly or manifest during or after adolescence. We know there's a great burst of myelination going on in the brain in the years between about 12 and 18. In fact, our brains are continually myelinating. My brain is still myelinating. Time course of symptom onset of mental illness and treatment effect is consistent with myelination changes. Now I'm just going to show a few slides. This is work done here at Adworth, Andrews et al. 2017, looking at the grey matter boundary and how fuzzy it is in subjects with autism spectrum disorder. There are 98 adults with ASD and 98 typically developed controls. The contrast measured as percentage between grey and white matter tissue signal at the grey-white boundary. Relatively low resolution scans, I think 1 millimeter or maybe 1.5 millimeter scans. So they're not really sampling the cortex very finely. But they found that ASD subjects had significantly reduced grey-white matter contrast in several clusters throughout the cortex, and there's a picture. So they suggested this as an in vivo proxy measure of atypical cortical microstructure. Another study of autism is done by Chen et al., published in 2022, looking at toddlers and preschoolers with autism and with ASD. What they found was that their T1 weighted over T2 weighted maps were largely comparable with normals. And so there were no group differences, but the development with age was different between atypical and ASD children in several early myelinated regions. In normal children, estimated myelin content increased with age across toddler and preschool years, but not in children with age. So there's another smoking gun. This is now work done at the Spinoza Center in Amsterdam, where I used to be a honorary professor. And they looked at major depressive disorder patients and healthy controls, and they found that it was areas in the inferior or the orbitofrontal cortex, shall I say, lateral orbitofrontal cortex, which was less myelinated than normals, using T1 as the index of myelin. And then finally, this is one done more recently here, published in Nature Communications, with Ed Wilmore as one of the co-authors and a lot of others from the imaging neuroscientists here, showing that resilient psychosocial functioning is associated with ongoing cortical myelination and functional reorganization during adolescence. And they find similar set of areas in the prefrontal cortex, which which myelinates more in these resilient subjects than in the non-resilient subjects. Adolescents who showed increasingly resilient response to psychosocial stressors, where they showed a higher rate of myelin-sensitive MT change, the anterolateral prefrontal cortex. So this is now just a set of questions. Can high-resolution quantitative structural MRI at ultra-high field show the bands of Baillet in many cortical areas and thus provide an extensive structural fascination? Does cortical myelination indeed proceed synchronously within each cortical area? Can this technique track intracortical development chains in myelination? Do onsets or bursts of myelination in specific cortical areas coincide with transitions in cognitive development? Can components of the feature spaces revealed by machine learning analysis of functional imaging studies, such as voxel encoding or representation similarity, be associated with specific myelin-defined cortical area? Okay, that would be illuminating the hypothesis that each cortical area has its own algorithm or set of algorithms. Can the hard wiring defined by the myelination pattern of each cortical area be used to model the operation of that area's particular microcircuits? Can models based on such area-specific microcircuits predict the observed feature space components of that area? Are slow developmental disorders such as autism and schizophrenia typically characterized by specific patterns of abnormal cortical myelination? We're beginning to see evidence that they are. Thank you for your attention.